welcome to the video for 6.2. So we just got done with this section 6.1, which was a kind of a beast. So in 6.2, we're going to continue talking about how we grow bacteria in the lab. So we're going to talk about cultures, and we're going to talk about bacterial growth. And specifically, we're going to talk about the growth cycle. So let's get started. Okay, so we're going to start with a review question. So review question number 1 says, the addition of sheep blood to agar produces a very rich medium called blood agar. Do you think blood agar can be considered a selective medium? What about a differential medium? Or could it be both? Okay, that's always an option. Okay, so there is a hint for this question. It says that some bacteria can lyse red blood cells while others can't. Okay, so take a second, pause the video. Now, I will say you do have the answer to this question on the next slide, but try to answer it on your own. Okay, so the answer to this is it is a differential media, but it's not a selective media. So just a reminder, selective means that we select or we allow only one type of organism to grow, okay, while either restricting or killing other types of organisms so they can't grow. Okay, and differential is a way that we can tell the difference between several different types of bacteria. Okay, so the blood agar is differential because we can grow different species on it because different species have different abilities to lyse red blood cells. Some can partially lyse, some can completely lyse, and some can't lyse them at all. So if you remember back into 6.1, we talked about the Haemophilus influenza bacteria that causes meningitis. So we showed the sheep blood agar and we showed the chocolate agar. Now, it didn't grow on the sheep blood agar, the blood agar, but it did on the chocolate agar. And that's because Haemophilus influenza needs stuff that's in the agar with the partially lysed cells, so the chocolate agar, but it can't lyse the cells by itself, which is why it didn't grow on the blood agar. So that's kind of what we're seeing here. So it's differential because we can tell the difference between the different species that are able to lyse blood cells or not, or partially, but it's not selective. It doesn't restrict the growth of anything. Okay, it's considered a rich medium. Oh, where's my thing? Okay, so this statement right here, very rich medium, means that it can't be selective because rich media is going to grow everything like we talked about in the last section and in the lab. Okay, this is why it's not considered selective. Okay, so let's talk about this because this is really important to know the difference between the two and to be able to do an example problem like or problem like the one on the previous slide. Okay, so selective versus differential. This is one of the things that I have a lot of students that have problems with. Okay, remember, selective media is going to select for the growth of a desired organism. Okay, which means it's going to inhibit means stop the growth of other types of bacteria, the non-desired ones, so the ones that we don't want to grow. It's going to stop them from growing or kill them altogether. Okay, differential media, on the other hand, isn't going to stop the growth of things, but it's going to have an indicator in it that's going to allow us to look and tell apart different species of bacteria. Okay, we're going to take advantage of some of the metabolisms of these bacteria, so the biochemical properties they have. Okay, so we will see a visible, usually a color change when we're growing these bacteria. Okay, so differential media does not kill anything. It does not stop growth. Okay, it's going to lead to a color change. Okay, selective media can stop growth. It can kill organisms. This is the biggest difference between the two. You have to know these two for the exam. Okay, so let's try our hand again. Let's try another example problem. Okay, we have enteric agar. So we call this HE agar. Okay, this is an agar that's used particularly for the isolation of Shigella species. Okay, so HE agar has bile salts incorporated into it, which are going to inhibit the growth of gram-positive bacteria It also has salicin, sucrose, and lactose that are fermentable carbohydrates. So depending on the bacteria, the colonies that can ferment these will either be yellow, orange, or salmon colored, depending on which of these carbohydrates they ferment. Okay, it has bromothiamyl blue and acid fusion that serve as acid-base indicators. Okay, so bromothiamyl blue turns yellow in the presence of acid. So do you think HE agar can be considered a selective medium, a differential medium, or both? Okay, so take a minute, look at the previous slide, and see if you can find the answer. Okay, so this is a long question, but it's actually really easy. Okay, so the answer to this example problem is that it is both selective and differential. So let's look at the parts of the question that tells us the answers. So the first part is whether it's selective or not. Does it stop the growth? And it literally tells us that it uses bile salts to inhibit the growth of gram-positive bacteria. It kills something. It stops growth. This means it's selective. Okay, so that one's done. So let's look at what tells us whether it's differential or not. Okay, so remember, differential is going to use an indicator or a color change to show the difference between different species. Okay, and it says right here in the next sentence that it has these three carbohydrates that will change the colony's color based on which one they ferment. Okay, so that's differential. It's a way to tell the different ones apart. So if one does salicin, it's going to turn yellow. If it does sucrose, it's going to be orange. If it's going to be lactose, it's going to be that salmon pink color. Okay, but it also has the different indicators. So if it does ferment something, it's going to produce lactic acid, and lactic acid is going to cause the bromothiamyl blue to turn yellow. So we have two ways that we can indicate or differentiate the different species from each other. Okay, so this is both selective and differential. I would know how to do a question like this for the exam. Okay, let's do one more review question and then we'll start on the lecture for 6.2. Okay, so review question number 2 says, you are interested in determining the number of viable cells in a bacterial culture. Which method of counting would you choose for the most accurate results? So A, spread plate dilution. B, direct cell counts using a microscope. This is like the fluorescent microscopy example we saw in the previous section. C, looking at the tube. Or D, optical density with a spectrophotometer. Okay, so pause the video, take a second, flip through your notes, and see if you can find the answer. Okay, so the answer for this one is A, spread plate dilutions. Now, the way to go about this question is you need to know what viable means. So if you remember correctly from 6.1, viable means that these are living cells. They're able to grow and reproduce. Okay, this is why A is the answer, because spread plate dilutions depend on growth. Okay, if we don't have viable cells, we don't see any growth. Okay, so we only see viable cells with this one. So B, direct cell counts using a microscope like in fluorescent microscopy, we see both the living and the dead cells. So it's not just the viable cells. Okay, looking at the two, well, we can tell if we have growth, right? We can look for how cloudy it is. But that doesn't tell us which ones are living and which one's dead. Because you can have a sample. I usually have one during the semester where I forget to feed the culture and I kill all the cells in the culture. Okay, so all the cell debris, all the dead cells are going to land at the bottom. So if I shake it, it's going to look cloudy. It's going to look like there's living cells, but they really all are dead. Okay, so looking at the tube is not going to tell me which ones are viable. Okay, and then the same with optical density, right? Because optical density judges how cloudy your sample is. Okay, it can't tell you how many living and how many dead there are. Okay, so this is an indirect way of knowing. This is a direct way, but we're counting both living and dead. And this one is the only one that really depends on viable cell growth. So that means we're only looking at viable cells. So the answer is A. Okay, so let's start into 6.2. This is all about the growth cycle. Okay, you need to be able to understand the phases of a typical bacterial growth curve, and you need to explain how bacterial growth correlates to disease. Okay, this is not a very long section. It's going to go pretty quickly, and it's not too hard. These objectives make it seem harder than it is. Okay, so let's start with the growth cycle. Okay, all living things have one ultimate goal. Okay, all living organisms have the goal to reproduce and have offspring and pass their genetics on to the next generation. That's what all living things are trying to do. Bacteria and microbes are the same. Their ultimate goal is to reproduce. Now the thing is, when microbes reproduce, they have both phases of growth where they can divide and make more bacteria, and they have periods of non-growth. Okay, we've talked about how some bacteria create endospores because the conditions are bad. So that would be a period of non-growth. And periods of growth is when they're in good environments and they have all the nutrients they need to grow and divide and just be happy. Okay, most bacteria are going to reproduce by binary fission. Okay, remember this is when one parent cell divides into two to make two identical daughter cells. Okay, the thing is, what you don't really talk about is binary fission can be symmetrical or asymmetrical. So we really have concentrated so far on symmetric binary fission. So the septation happens in the center. It divides equally. Okay, so this is symmetrical binary fission. Now we can also get things like we see in yeast. So we have this cell that's budding off the other. Okay, it's creating this stalk in between. So what we have is when the stalk breaks, one cell is going to have the remaining piece of the stalk, and they're going to be oddly shaped. They're not going to be the same size. This is what we mean by asymmetrical binary fission. Okay, we don't see a lot about this, so we're probably not going to see much of it for the rest of the year, but just something you should know is that everything's not symmetric when it divides. Okay, and then we have eukaryotic microbes, because binary fission happens mostly with prokaryotes. Eukaryotic microbes, they divide by mitosis, and we're going to talk about mitosis in a lot more detail in a later chapter, just not right now. Okay, so binary fission, so bacterial reproduction is going to lead to exponential growth because when bacteria divide, that one cell is going to double every time. So we say it doubles at a fixed rate. This is what leads to exponential growth. So if you remember from math what an exponential curve looks like, let me see if I can't write on this. Okay, we're going to try this. So we're going to have our axes. Woo. Okay, so we're going to start at a constant number of cells. So this could be like 1. So it's going to stay constant for a little bit, and then all of a sudden, it's going to really jump. Okay, so it's going to go, whoo. This is going to be an exponential growth curve. Okay, so because bacteria like to reproduce at a fixed rate, so say like every 20 minutes, that means they're doubling every 20 minutes. So we can go from 1 cell to 2 in 20 minutes, from 2 cells to 4 in 40 minutes. And then in 60 minutes, we can go from 4 to 8. And then in 120 minutes, we can go from 8 to 16, etc., etc., etc. We're just going to keep dividing. Okay, because of that, each of those cells are going to keep dividing too. So they're going to divide into 2 and double it every generation. Okay, so this is in ideal conditions. We have to remember a lot of this lecture is going to be underneath ideal conditions. So in order to get exponential growth, we have to have plenty of nutrients. We have to have really good growing conditions. So bacteria depend on temperature, pH, nutrients, osmolarity, and a few other factors. But those 4 are our big ones. Okay, so temperature, a lot of pathogens like to grow around 37 degrees C because that's our body temperature. Okay, so 98.3 degrees Fahrenheit on average. Okay, so that is the perfect growing temperature for pathogens in our body. That's why they like us. Okay, you take that same pathogen and put it out on the desk, and you don't really see any growth. We'll see this in the lab. Okay, when we do the swabbing, you're gonna put one in the incubator and one on the counter, and what you should see is different growth in between the two, because different things grow at different temperatures. Now, a lot of bacterial species like to be in a pH neutral, so around 7. Okay, we always know the more food, the better for bacteria. But the one that we really don't talk a lot about is osmolarity. So this is like a concentration gradient, um, but it's going to be high salt concentration. So the more salt in the environment, the less likely the bacteria are going to grow. So we want low osmolarity. So we want low salt content in the environment, and that's going to allow the bacteria to grow better. Okay, so in ideal conditions, we're going to double at a fixed rate. But a lot of times, our bacteria aren't in ideal conditions. Nature has a lot of constraints and restricts things from growing. Okay, so in nature, bacteria very rarely have an exponential growth curve. Okay, so for us in the lab, we have exponential growth. We have a fixed generation time. Okay, also the generation time is also sometimes referred to as the doubling time. Okay, so we can have it double every 20 minutes. That same bacteria out in nature could take considerably longer than 20 minutes. It could go up to a full day longer to divide and replicate. Okay, and that's just because the conditions for growth are different when we do it in the lab because our cultures are formulated to be ideal. Okay, so on the previous slide, we talked about how bacteria can double every time they go through a generation. So that's what leads to that exponential growth. Okay, so we can easily know that one generation time takes one bacteria to two, then from two to four, from 4 to 8, and 8 to 16. So we can just do it doing that the normal way. But sometimes we get a lot of generation time, so it's easier to use an equation. So our bacterial growth equation is going to be the number of organisms is equal to N0. So N0 is the number of organisms at time 0, so how many you start with, times 2 raised to the N, where N is the total number of generation times you're letting the bacteria grow. Okay, so for example, one way we can use this equation is a single cell after 3 generations, that means that N is going to be equal to 3, will produce how many cells? Okay, so we can use our knowledge of how things double. So we know it's going 3 generation times. We're starting with 1, so 1 goes to 2, 2 goes to so 1 goes to 2 after 1 generation, 2 goes to 4 after 2, and 4 goes to 8 after 3 generations. Or we can use this bacterial growth equation. So we know that we have 1 cell at time 0. We're going to go through 3 generations. So this is going to be 1 cell times 2 raised to the third, and 2 times 2 times 2 is 8. So we get 8 cells. So either way you decide to do it on the exam and on the homework, you are going to get the same answer. It's just sometimes a little bit easier with the equation versus just doubling it. Okay, so we've talked about exponential growth. We've talked about generation time. So what we have here in this picture is a picture of what we call a batch culture. So our batch culture is going to be in an Erlenmeyer flask. It has a specific shape that allows for swirling. Okay, we have a big batch, hence batch culture. Okay, so we're going to see how this grows. Okay, remember, batch culture is an ideal condition. So we have perfect temperature, we have perfect pH, we have the excess nutrients, and perfect osmolarity. Okay, so because we can swirl these are actually on what we call a shaker, and it does exactly what you think it does. It literally rocks back and forth and shakes the flask. So what's happening is the broth is constantly rotating, which is constantly moving the bacteria and constantly stirring up the nutrients. So it allows for more even growth in the flask. Okay, so we can use a batch culture to look at how bacteria grow in culture. Now, these are ideal conditions. They're not always realistic because nature is different than in the lab. But we are going to look at how bacteria will grow in this batch culture. Okay, so this next slide is something we really do need to know. Okay, so this is what we call the bacterial growth curve. Okay, so this happens in batch culture. So one other thing that we should talk about is with this slide, or with this curve, it is in what we call a closed system, meaning we don't put anything else in after we start, and we don't take anything out. Okay, so it's closed. So we're allowed to see how things change as things progress. Okay, so our bacterial growth curve is going to have 4 parts. Okay, we have the lag phase here. We have the log phase. We have the stationary phase, which is 3, and 4, we have the death phase. Now, this is a logarithmic scale. That means every unit we have, so like this 10 to the 5th, is going to increase by a factor of 10 each time it increases. So we go from 10 to the 5th to 10 to the 6th. So this is 10,000 going, or no, 100,000 going to 1 million. So 100,000 times 10 equals 1 million. So we're going to jump by a factor of 10 by each unit on this scale. Okay, so what we're going to do is we're going to go through what happens at each of these stages. You need to know this growth curve and what happens in each one of these. You will be tested on this. Okay, so let's start with the lag phase. Okay, so the lag phase is when growth really doesn't happen. It stays constant. Okay, so we have the same amount of bacteria throughout the whole lag phase. And then at the end, they're going to start to grow. Now, there's a bunch of different ways or a bunch of different reasons why bacteria don't grow immediately. So the first part could be is the bacteria have to sense that they're in a good environment. Okay, they have to make sure that everything's there so they can reproduce and grow. So they're going to do like we talked about in the last chapter, and they're going to use their receptors. They're going to start to taste around the environment, and when they realize it's a good environment, that's when they're going to kind of flip the switch and say, "Oh, I can grow." Okay, sometimes they need to realize they're in a good place. Sometimes they need to prepare for growth. Maybe they need to collect nutrients. Maybe they need to be a little bit bigger before they divide. Maybe they need to replicate their DNA. Okay, and sometimes they have to fix damage. Okay, so sometimes I'm a little bit lazy and I wait the longest amount of time to start a new broth culture. So in old cultures, our cells might be slightly damaged. They're still viable, they're still living, but they're slightly damaged. So when I transfer them from an old culture to a new culture, before those bacteria can start growing, they have to repair themselves in order to grow again. So we have slightly longer lag phase than with a bacteria that just is sensing a new environment. Okay, so like when they enter a new host cell. Okay, so the lag period can be short or long depending on the new medium, the new location, or what shape the cells are in when they get into that good environment. Okay, so my really damaged cells from my culture take a little bit longer to start to grow. They have a longer lag period. Okay, I would know what happens in this phase. Okay, so the next part is called the log phase. Sometimes this is referred to the exponential phase. This is where we get maximum growth, right? We hit that point, our cells are ready to divide, and then they start dividing. Okay, so we start doubling. So we're going to have our exponential part of our curve here. Okay, so we're growing, we're doubling. The amount of cells in the culture is rising. So we're getting more and more cell dense. Okay, so as density rises, more bacterial cells are going to be in the culture. Okay, so we're going to get to a place towards the end of the log phase. So somewhere around here, we call this late log phase. We have so much bacteria, they're starting to bump into each other. There's going to be a drop in nutrients because everybody's eaten, right? We have tons of bacteria. Okay, so at the end of the log phase, the bacteria are going to start to communicate with each other. Okay, right? They're bumping into each other. They know they're there. We call this act of communicating with each other quorum sensing in the late log phase. Okay, I heard Dr. Burrows, who is our liaison for this class, refer to it as Twitter for microbes, and I thought that was a really great analogy. Because in quorum sensing, the bacteria are going to start sending messages to each other. So one's going to send one off to each other, the other one's going to reply back. So they're going to be sending and receiving constant signals and being in communication with each other. This all happens in the log phase. Okay, so the stationary phase, we have pretty much exhausted all resources. We don't have very many nutrients left. We have so many bacteria that have been dining on these nutrients. They have produced waste and shipped it out of them. So the the culture is kind of becoming a little bit toxic because we have so much waste built up. Okay, so in the stationary phase, we really don't see any growth. we've plateaued, like literally plateaued in the graph. Okay, so we're not growing, but we're not dying yet. Okay, so a lot of the times the stationary phase can be extended by the fact that some bacteria can literally change their physiology to deal with the fact that there's no more nutrients or there's a lot of waste products. So E. coli, for example, when they get to the stationary phase, they change their size. They become smaller so they don't need as many nutrients so they can survive longer. Okay, other species like Bacillus anthracis, they're going to develop endospores. Okay, they're going to sense in the environment that there's no nutrients in their environment and that there's lots of waste products, and they're going to be like, cool, I'm out, right? It's going to shove all of its genome into an endospore and wait until conditions get better. Okay, this is why it plateaus here at the top, is because nothing's really dying yet, but nothing's growing either. We're not going through binary fission anymore. Okay, so the fourth stage is the death phase. Okay, so this is when our cells start to die off. Now, one question I get is like, Miss Harker, why is this not exponential? Like, our growth is exponential. Why aren't they dying as fast as they were growing? Well, there's a few reasons for this. Okay, sometimes bacterial cells will get some mutations from the toxic environment that they're in, and this is going to give them abilities that are going to allow them to survive in that toxic environment while other cells are dying off. Okay, and some cells adapt pretty quickly, and they realize that they can eat other cells. So they're literally going to cannibalize other cells. So the death rate, so how fast the cells are dying, is going to be pretty gradual. So it's going to just slowly decline because some of these bacteria have adapted to prolong and put off their deaths. Okay, so we don't see a steep drop off in the death rate because of this. So it's not as steep of a curve as when they were growing. Okay, so that is section 6.2. We've now talked about the growth cycle. So you know the lag phase, the log phase, the stationary phase, and the death phase. And we've talked about the different ways that bacteria can change their attributes through the different phases. And we've talked about how growth is exponential. We know how to calculate it now. We know about batch cultures. Okay, so in 6.3, we'll come back and we'll talk about what we actually feed bacteria in order to get them to grow. So see you in the video for 6.3.